Hello, welcome to Beyond Science. In today's video we will see how light intensity affects the rate of photosynthesis. Plants are known as producers because they use things from their environment to make organic compounds. Plants take in carbon dioxide from the air through stomata in their leaves. The roots of a plant absorb water from the soil. The pigment chlorophyll within chloroplasts absorbs sunlight. Plants use these things to produce glucose and oxygen. How quickly photosynthesis happens depends on the availability of carbon dioxide or chlorophyll, on the temperature and on the intensity of the light. Here is a hypothesis. The rate of photosynthesis depends on light intensity. What are the variables in this investigation? How could we measure them? We're going to change the light intensity. This will be our independent variable. We will change the light intensity by changing the distance between the photosynthesizing plant and the light source. We're going to measure the rate of photosynthesis. This will be our dependent variable. To do this, we will observe how much oxygen is made by photosynthesis in 1 minute at each level of light intensity. Since temperature and the availability of carbon dioxide can also affect the rate of photosynthesis, we will need to keep these things the same during our experiment. Temperature and the availability of carbon dioxide are control variables. To set up this experiment, we need a sample of pondweed or other aquatic plant. Trim the stem of the pondweed, then place the pondweed in a large beaker full of water. Add some sodium hydrogen carbonate to the water. This ensures that there will be plenty of carbon dioxide available for the pondweed. Place a glass funnel on top of the pondweed and add a boiling tube full of water over the top of the funnel. As the pondweed photosynthesizes, it will release bubbles of oxygen gas. The oxygen will collect at the top of the boiling tube. Place a 1 metre ruler on the work surface and position a lamp at the 0 centimetre mark. It is best to use an LED lamp if available because they do not become hot. If you don't have an LED lamp, you can use a tank or another large beaker of water as a heat shield. This will prevent the water around the pondweed becoming hotter and the temperature affecting your results. Position the edge of the beaker containing pondweed at the 20 centimetre mark on the ruler. Switch the lamp on and allow the pondweed to acclimatise for 2 to 3 minutes. Start a timer and count the number of bubbles of oxygen that the pondweed produces in 1 minute. Move the beaker with the pondweed so that its edge is in line with the 30 centimetre position on the ruler. The plant is now further away from the lamp and is experiencing a lower light intensity. Once acclimatised, count the number of bubbles of oxygen produced in 1 minute. Move the pondweed to the 40 centimetre position on the ruler and count the number of bubbles made in 1 minute again. Repeat this at 50 centimetres. and at 60 centimetres. You should repeat the test and calculate a mean at each distance. How does changing the distance between the lamp and the photosynthesizing plant affect the rate of photosynthesis? As the distance between the pondweed and the lamp increases, the light intensity decreases and the rate of photosynthesis also decreases. We can see this because as the pondweed was moved further away from the lamp, the mean number of bubbles of oxygen produced in 1 minute decreased. How could this investigation be improved? You may encounter some difficulties when carrying out this method. For example, the bubbles might be produced so quickly it is difficult to count them, or that the bubbles of oxygen can vary greatly in their size. You might also find that bubbles are released from various parts of the plant and you might not see every bubble. To improve this method, we could use a gas syringe to accurately measure the volume of oxygen that is produced in 1 minute at each level of light intensity. If you have enjoyed this video, don't forget to like, subscribe and ring the bell. You can also check out other videos in this cell biology series, such as cell specialisation and differentiation, mitosis in the cell cycle, and transport in cells.